Hello and welcome everybody to my channel Omni Bioentrance, and today I will be discussing IISER Aptitude test or IAT conducted in the year 2022. I will be discussing only the biology section. If you are new to my channel, please do like, share and subscribe. Coming to the first question. 1 minute. Okay. Match the features of a plant cell listed in column 1 with their corresponding examples in column 2. This is column 1. Column 1 cell tells cells with cytoplasm and no nucleus. So it is mature sieve tube. Mature sieve tube doesn't have nucleus, but it has cytoplasm. Then next one, cells lacking both cytoplasm and a nucleus. It is R, that is trachea. Trachea is a dead cell, so it doesn't have both cytoplasm and nucleus. Then third one, cells containing more than 2 nuclei. So it is 3. It is tapetum. It contains more than 2 nuclei. Haploid cells produced by mitosis. That is pollen grain. Haploid cell produced by mitosis is pollen grain. Answer here is option A, that is 1T, 2R, 3S and 4Q. Then next one. Shown below are some of the reactions that occur in the metabolic pathway leading to complete oxidation of glucose during aerobic respiration. That is your glycolysis. As well as Krebs cycle. That only complete oxidation of glucose is taking place during aerobic respiration. So you have to choose the correct sequence of reaction during the complete oxidation of glucose. So if you see here, first one will be dihydroxyacetone phosphate to glyceraldehyde 3-phosphate. That is in your glycolysis case. Then pyruvate to acetyl-CoA. Again after glycolysis, that is after the production of pyruvate, it will get converted to acetyl-CoA to enter into the Krebs cycle. Then you have to see further steps in the Krebs cycle, which are 3 and 4. So which one is occurring first? Oxaloacetate to citrate, then fumarate to malate. So the answer here will be 2, 1, 3 and 4. Answer is option A. Coming to the next question. In a given species of flowering plants, the color of the seeds is exclusively determined by the color of its seed coat. Seed coat color in this species is governed by a nuclear gene with 2 alleles. The white, capital W allele is dominant over the brown, small w allele. If a plant with brown seed, small w, is crossed as a female with pollen from a white seed, capital W plant, what will be the seed color and the genotype of the embryo in the resultant seeds obtained from this cross? So this is just a very simple genetic problem, and it's very easy to do. So this is the female plant which is brown seeds, and it is crossed with a pollen which is white seeds. So small w will be the gamete of female and capital W will be the pollen. Crossing you will get capital W, small w. So W is dominant, so it is white. So white seeds with WW embryo. Capital W, small w embryo. Answer is option B. Then next one. During the COVID-19 pandemic, SARS-CoV-2 virus mutated multiple times, giving rise to many variants. What is the genetic material of the SARS-CoV-2 virus? So it is single-stranded RNA. The genetic material for SARS-CoV-2 virus is CoV-2 virus is single-stranded RNA. Answer is option A. Lymph is an important body fluid. Choose the incorrect statement about the lymph. It remains as an interstitial fluid and is never put back into circulation. It is the interstitial fluid generated by the passage of liquid between the cells of the capillary. It is colorless and has similar mineral composition as of plasma. Fat digested in the intestine is absorbed through the lymph. So B is correct, C is also correct, D is also correct. So what is not correct is A. So your answer here is option A, because they have asked the incorrect statement. And before explaining that, I will explain A. So what is interstitial fluid? Tissue fluid is also known as interstitial fluid. It is the fluid running between cells and blood capillaries in a tissue or an organ. The tissue fluid, when entering into the lymphatic system, it is known as lymph. So it remains as an interstitial fluid and is never put back into circulation. Lymph is put back into the circulation. It doesn't remain as an interstitial fluid always. Okay. So the incorrect statement here is option A. Option A is your answer. Then next one. Interneurons play an important role in the execution of spinal cord mediated reflex. Where are these interneurons located? So interneurons, they are located in the gray matter. Answer is option A. Then next one. Osmoreceptors are sensitive to changes in ionic concentrations and volumes of body fluids. Yes, because when osmoreceptors detect changes in the concentration of ions or volumes of body fluids, they send signal to the hypothalamus, because osmoreceptors are located in the hypothalamus, where they triggers a response to restore the balance. Now, which among the following best describe the function of these osmoreceptors? They stimulate hypothalamus to facilitate the release of antidiuretic hormone and increase water reabsorption. So, if there is, suppose, if there is, or for example, I can say, if the concentration of ions in the blood becomes too high, then these osmoreceptors will sense that. They will send the signal to hypothalamus. Hypothalamus will send signal to pituitary gland to release ADH, which causes the kidneys to retain water and reduce the concentration of ions in the blood. Or it will cause the kidney. Yes, that's correct. So the answer here is option A. They stimulate hypothalamus to facilitate the release of antidiuretic hormone and increase water reabsorption. So answer is option A. Then next one. A single point mutation in a gene X results in breathing difficulty, hypertension as well as partial sterility. Which among the following phenomenon best explains the observed phenotypes? So it is pleiotropy. In pleiotropy, one gene influences two or more phenotypic traits. So here the gene X, it influences breathing, hypertension and partial sterility. So if there is a mutation in this gene X, it results in all these problems like breathing difficulty, hypertension as well as partial sterility. So it is an example of pleiotropy. Then next one. In a cross between individuals of the genotype PPQQRRSS and PPQQRRSS, what will be the expected number of progenies with the genotype small p, capital Q, capital R, small capital S in a population of 400 individuals assuming independent assortment? So this is the cross between these genotypes, that is this genotype and this genotype. What will be the number of progenies with the genotype this? what they have given in the question. I am not telling capital P, small p, capital Q. It will take a lot of time. You can see it over here in the question. So when you cross capital P with small p, that is these two, P, capital P, small p with small p, what do you get? You get, these are the gametes obtained, and you get capital P, small p with small p, small p. Okay, but in the progeny genotype, it is both small p, small p. Okay, okay, you will get small p, small p. So we can take that into consideration. Again, when you cross capital Q with small q, small q. So capital Q, small q, small q. So capital Q, small q, these are the gametes, and you will get capital Q, small q. But here in the question, they have asked for the genotype where both are capital Q. That will not be obtained. So the genotype given in the question cannot be obtained. So what will be the expected number of progenies with this particular genotype will be 0. Answer is option A. Then next one. Organisms in which of the following phyla are triploblastic, acoelomate and have bilateral symmetry? So they are platyhelminthes. Answer is option A. Then consider the following biomes: tropical rainforest, tundra, desert and coastal zone. Choose the most probable descending order of net primary productivity of these biomes. So what is net primary productivity? The amount of energy that remains after respiration loss in the producer is referred to as net primary productivity. That is, if you didn't explain, I will try to simplify it. That is the amount of energy the plant captures through photosynthesis minus the energy they spend for cellular respiration. That is net primary productivity. Now you have to see for the descending order of net primary productivity. Now if you take tropical rainforest, it will have the highest net primary productivity because it is rich in biodiversity. It has got abundant sunlight and consistent rainfall. So it is having the highest net primary productivity. That is the amount of energy the plant capture through photosynthesis minus the energy they spend for cellular respiration will be maximum in tropical rainforest. Then next, if you see the coastal zone will have net primary productivity that is lower than the tropical rainforest because of their variable environment of the coastal zone. Why the coastal zone is having variable environment? This may be because of temperature and wave action that can cause a variable environment to form in coastal zone. So the next will be P greater than S greater than, the next will be tundra. Tundra ecosystems, they have still lower net primary productivity due to their extreme cold temperatures and limited plant diversity. Similarly, desert also has net primary productivity which is lower than that of tundra because of the arid conditions and very sparse vegetation. So because of all these reasons, the descending order will be P greater than S greater than Q greater than R. Answer will be option A. Then next one. 9% of a population cannot taste a certain food item because of the recessive allele of a gene IAT. Assuming the population is in Hardy-Weinberg equilibrium, what will be the frequency of dominant and recessive alleles respectively? So you have to see for the value of P and Q here. P will be the frequency of dominant allele and Q will be frequency of recessive allele. Q they have already given in the question as 9%. So Q square is equal to 9%. 9 by 100, Q will be 0.3. Not Q square, Q will be 0.3. Then P plus Q is equal to 1. We know as per the Hardy-Weinberg equilibrium, P is equal to 1 minus Q or 1 minus 0.3, that is 0.7. So the answer here will be, they have asked for the dominant and recessive allele. Q is the recessive allele because they have already mentioned 9% of the population cannot taste a food item because of a recessive allele. So Q is the recessive allele and P will be the dominant allele. So 0.7 and 0.3. So the answer here is option A. Then next one. As opposed to DNA replication within the cell, discontinuous synthesis of DNA does not occur in a polymerase chain reaction. Why? Now in normal cells what happens is the DNA replication is bidirectional, where replication forks move in opposite direction from the origin of replication. And each replication fork, it has got a leading strand that is synthesized continuously in the 5' 3' direction, and another strand which is called as the lagging strand, which is synthesized discontinuously in short fragments called as Okazaki fragments. DNA polymerases in the cell work unidirectionally, adding nucleotides only in the 5' to 3' direction. This is what happens in normal cellular conditions. Here they have asked, as opposed to DNA replication within the cell, discontinuous synthesis of DNA does not occur in a polymerase chain reaction. Why? Why there is no discontinuous synthesis of DNA in a PCR? I already told you that in normal replication, discontinuous synthesis take place as in the form of Okazaki fragments. But in PCR, no Okazaki fragments are formed. Why? So, if you see the case of PCR, the primers are designed to anneal to the template DNA in opposite direction. See, this is the DNA template, 5' to 3' and 3' to 5'. This is the forward primer. Usually you will think the forward primer will bind over here and the reverse primer will bind over here. No. The forward primer will bind to 3', 5' strand and the reverse primer will bind to the 5', 3' strand. Okay. So this is the forward primer binding. This is the reverse primer binding. And they will allow the synthesis of the DNA strands in each direction, that is in the 5' to 3' direction. The synthesis will go like this because you add nucleotide to the 3' end. Here also it will be synthesized in the 5', 3' direction because nucleotides are added to the 3' end. So the synthesis will be in 5', 3' direction. So the first option, if you see, denaturation step in PCR substitutes for the replication fork. Yes, here the DNA is denatured due to high temperature in PCR. So it doesn't take into consideration regarding origin of replication or anything like that. There is denaturation due to high temperature and the strands separate. The primers anneal, forward primer anneals here, reverse primer anneals here. And the strand, newly synthesized strand will be in 5', 3' direction. So first option, denaturation step in PCR substitutes for the replication fork and Taq polymerase extends DNA only in 5', 3' direction. So the answer here is option A. Then which of the following figures correctly represent the chemiosmotic hypothesis of ATP synthesis occurring in a mitochondrion in cell? So I have given only one option, that is option A, that is the correct option here. The other options I have included in the next slide. They are all not correct. B, C, D are not correct. Only A is correct. Why I will tell you. So electrons carried by NADH. See here. Electrons carrying electrons from food. NADH or FADH to from glycolysis formed during glycolysis and Krebs cycle feed the electron carriers. These are the electron carriers in inner mitochondrial membrane. This is the inner mitochondrial membrane. These are the various electron carriers. These NADH and FADH2, they will feed the electrons to various electron carriers of the inner mitochondrial membrane. As a result of this, they will pump protons to the intermembrane space. This is the intermembrane space, this yellowish shade. They will pump electrons to the, they will pump H positive ions to the intermembrane space from the matrix. See here, H positive ion from matrix to the intermembrane space. See the arrow, direction of the arrow. From the matrix to the intermembrane space. From the matrix to the intermembrane space. These are the various electron transport complexes. There is complex 2 also. They have not shown it here. Maybe because it is not involved in transport of pumping of H positive ions to the intermembrane space from the matrix. Then this proton pumping, it will create an imbalance of H positive ion. Thus a charge difference between intermembrane space and the matrix. The imbalance is the proton motive force. The proton motive force drives proton back to the matrix. This imbalance, it will drive the H positive ions back to the matrix through the ATP synthase. That is FC. This is the ATP synthase. This is the F0 subunit. This is the F1 subunit. And the H positive ions enter from the intermembrane space to the F0. Then it will move to the F1 and it will be transported to the mitochondrial matrix. This movement of protons is coupled to the formation of ATP from ADP and Pi by the F1 unit, not the F0. So from the F1 unit. So if you see this IMS is intermembrane space, the inner mitochondrial membrane, and this is the mitochondrial matrix. This is the electron transport complex. This is the ATP synthase. See here, it is the ATP synthesized by the F1 of the ATP synthase complex. F0 is through which H positive ions enter the matrix. So A is the correct answer. And here H positive ions move from the matrix to the intermembrane space. See the direction of the arrow. If you see the other options, you can see that ATP synthase is placed the opposite direction over here. ATP synthase, the F1 complex is facing the inner membrane space. In option B, it is facing the inner membrane space. That is not correct. Now if you see option C, the hydrogen ions are moved from the mitochondrial matrix to the intermembrane space, which is not correct through ATP synthase, and it is from intermembrane space to the matrix by electron transport complex, which is not correct. Then similarly in the case of D also, ATP synthase is placed in the reverse direction or opposite direction. So D is also not correct. Now next one. Let X be the perpendicular distance from the centromere of each chromosome to the equatorial plane of a human cell. Which of the following stages of the cell cycle is most likely to have the highest average value of X? So X is the perpendicular distance from this is the centromere of each chromosome to the equatorial plane. This is the equatorial plane of a human cell. So if you see the metaphase plate, the chromosomes will be arranged on the equatorial plane. So X will be equal to 0. But if you see the late anaphase stage, the chromatids have got separated because of spindle formation and they are pulled by the spindle fibers towards the pole. So the perpendicular distance from the centromere of each chromosome to the equatorial plane, that is this, from the centromere of each chromosome to the equatorial plane, that is x, that will be highest in the case of late anaphase stage. But in the early anaphase, they have given C, early anaphase stage, the chromatids split but move very little. So X is very small. So the answer here is late anaphase. Answer is option A. So thank you for watching my channel. Please do like, share and subscribe. Thank you.